Okay, good morning, everyone. Welcome to our second class. I wanted to point out, earlier this morning when I was giving my Bio 275 lecture, there seemed to be some problems where when I was moving through the slides on my PowerPoint, that the students were not able to see the slides changing, even though on my screen they were changing fine, which is really annoying, of course, because it means I keep talking about some slide that the students aren't even seeing. And I think this actually happened part of our first lecture on Monday with this class too, although I wasn't sure what it was that I was actually seeing. So I think there might be some weird glitch in the newest version of Zoom that I downloaded. Because like a genius, I thought, oh, it's a new semester. Let me see if I need to update my Zoom. And I updated it. I probably should have just left it. Like, if things aren't broken, don't try to fix it, right? So anyhow, if during the course of today's lecture, it seems like I've moved on to another slide, but nothing's changing, feel free, unmute yourself and just let me know, hey, Dr. Shen, I think this glitch is happening. And I think I can just kind of go back and forth in the slides to make it kind of catch up. All right. The poor Bio 275 students, they were all just like sending me chat messages, right, which I don't even see until the end. So when I ended the lecture, I was all excited because there's like 24 chat messages. I thought they were asking all these great questions, but all of them were like, I don't see the slide that you're talking about. All right. Okay, anyhow. So hopefully I will fix that. I'll fix that as soon as I can and figure it out. Hopefully you saw in La Lima, I made an announcement that let you know that the first homework assignment is available. It was available after Monday night, and it is due this Friday at 11:59, I think, basically midnight, Friday night, right? So it is in La Lima under assignments, tests, and assignments, tests, and surveys, I think is the tool. And it's just 4 short answer questions that have you think about some of the important things we've talked about this week so far. All right. So it's not meant to be a super difficult thing, but it is meant to make sure that you're thinking about the things that we're discussing in class and some of the concepts. Right? Because as a 400 level class, as a capstone class, I expect that all of you are capable of actually critically thinking about the things that we are discussing and not just retyping things that, that I'm asking for definitions or something like that. Okay, so look for that homework assignment. You can do it anytime between, now and Friday night, but don't forget to do it. All right. Okay. So let's move on and talk about tumors and how we categorize different types of cancers. Last time we left off with this idea that it's very important to have some sort of insights into what the normal body cell types are that are responsible for giving rise to a particular tumor that a patient is presenting with in the clinic. And we said that just like these crackers here, whereas if these are the normal cells of our body, and this is what we consider diseased cells, it's not necessarily clear which original normal cells gave rise as the origin to these diseased cells. And why it's important to know that is because certain cellular pathways or different types of signaling pathways in these cells may be different in terms of the types of pathways that are being emphasized and used by those normal cells when they were normal and well behaved. And there's a good chance that those pathways are somehow responsible for leading to this diseased phenotype. So if you have an understanding of what the original cell was, then you already have sort of a head start in being able to identify, for example, different types of therapeutic drugs that might target specific cellular pathways that are relevant for this type of cell, but maybe not relevant for this type of cell. Right? And that might save you valuable time in treating the patient. So it's a complex task, though, right, to identify the origins of these. And so this table in the book, I know this is a really scary table to start off with, and it gives you a sense of how challenging it can be to classify these different tumor types. Right? I don't expect you to know this whole table. I do expect you to understand how this table is put together. Right? You can see up in the corner here on the left, this yellow block is the main sort of breakdown of the criteria that's used to make or divide up all these different cancer types into these other colorful groups over here. Right, so these Roman numerals 1, 2, and 3 are what we are going to talk about today. Okay. So here they are. All right, so tumors are typically classified through this hierarchy, starting with differentiation state. Right? When we say differentiation, what are we talking about? Remember, the cells of your body that you see today, when you look in the mirror, right, typically are fully differentiated cells. They started off as stem cells somewhere in your body's tissues. You know, you have a population of what we consider multipotent stem cells that can continuously replenish things like the different cells that make up your blood, the different cells that make up your skin, et cetera, et cetera. If you can follow back and know what the original type of cell was that differentiated into something like skin or hematopoietic blood cells, right? That's an important categorization. So the three subcategories of that are epithelial, non-epithelial, and mixed. The second would be understanding where these cell types originated from way back when you were still an embryo, right? An embryo that, of course, started off as one fertilized egg cell and then began to divide to form a blastocyst, right? Essentially like a basketball of pluripotent cells, right? That then at some point got to the point where it started forming the different germ layers or different tissue layers that then defined all the different types of tissues in your body. So, for example, the primitive ectoderm or endoderm or mesoderm, right? That's an important part of understanding then the nature of these cells. And then the third is the biological behavior of these tumor cells that you're looking at. Are they benign or are they malignant? Okay, and let's go ahead and elaborate on these. We'll start off by looking at the differentiation state. Okay. So tumors arise from many different cell types. I mean, the truth is, any nucleated cell in your body has the potential to divide, right? Most of your differentiated cells won't, because that's not their job anymore. Their job is not division. It's doing the job that they're meant to do, like a nerve cell, a gland cell, a muscle cell, hair follicle cell, whatever, right? But the potential for them to divide is still there. So that means, by definition, that if a cell is still capable of dividing, it is also capable of losing control over dividing. So pretty much any nucleated cell in your body has the capability of becoming a tumor and eventually becoming cancer. So the three main categories of cell types that give rise to cancers are epithelial, non-epithelial, and mixed lineage. So let's start with epithelial. So epithelial cells are things like gland cells or the cell linings of different types of borders of like your mucosal membrane, your skin, etc. Right? And those give rise to the majority of human tumors. So epithelial cells are actually responsible for about 80% of all cancer deaths that we have in Western clinics. Right? The types of tumors originate from epithelial cells. Now, why is that? Let's take a closer look at epithelial cells and see if we can understand. Okay, so in this figure here, we have a really zoomed in image of epithelial cells. So here are epithelial cells. Epithelial cells in your body are typically associated on one side of them with a basement membrane. Right? And below that membrane on the other side is connective tissue. What you're seeing here are many of the collagen protein fibers that make up the connective tissue. Then you have this basement membrane, which is a very important feature as we learn about the progression of cancer. And then on top of the basement membrane sits these epithelial cells. Here's another cross-sectional image where you can actually appreciate that these are cells here. So this, if you can see, this sort of whitish boundary is the rightmost part of one cell. This cell is kind of squished in the middle, right? And there's another cell off to the right here. The actual basement membrane, you can see, just looks like this kind of wispy little line here. Right? And then below that is the connective tissue. Right? So epithelial cells, by nature, are constantly being turned over. Right? So the stem cells that give rise to epithelial cells are constantly busy at work replenishing things like all of the skin cells that you lose every day, right? Or the lining of things like your gastrointestinal tract, the epithelium that lines those are always being kind of sloughed off, either physically by food moving by them, right? Or just them getting old and falling off and being expelled from your body. Epithelial cells, by their nature, right, are the product of lots of proliferation. So that means the machinery inside epithelial cells is already kind of geared up for proliferation, right? Even though a fully differentiated skin cell or gland cell really isn't going to be dividing anymore. So therefore, there's greater potential, right, for mistakes in proliferation control to happen in epithelial cells. Another reason why epithelial cells are the most common source of tumors is because of their location. They are located in places in your body where they tend to be more exposed to things that can damage DNA. For example, your skin, right, is exposed to UV radiation that's constantly damaging their DNA compared to certain cells that are buried deep in your body where the UV rays cannot penetrate to. Likewise, the epithelial lining of our gastrointestinal tract is the first kind of barrier where all kinds of things that we're eating, including things like chemicals in our food, either natural chemicals or artificial chemicals like preservatives, right? Those cells are exposed to potential mutagens that way. Okay. So cancers that come from epithelial cells are called carcinomas. So this is very specific, right? If you hear the word carcinoma, you know that this is a type of cancer that came originally from some kind of epithelial cell. Many of these are from epithelial cells that are glands. So glands, for example, glands that secrete things like different substances in your body, right, inside of your organs that are creating different types of things that are released into your blood. These gland cells, if they actually start to get altered and proliferate out of control, right, they can form what we call adenocarcinomas. So adeno refers to a gland. Right? So if you see not just a carcinoma, but it's actually called an adenocarcinoma, you know that it came from an epithelial cell, and this epithelial cell was some sort of a gland cell. So just because a cell starts losing control and forming a tumor, that doesn't necessarily mean it's cancer. Because I think all of you are aware that you can have polyps, right, which are sort of localized little growths of cells that are maybe dividing more than they should, right? And they can form this little mass. But that little mass in itself is not cancer because that little mass can just stay like that, potentially indefinitely, right? So if you have a little mass like that, a little tumor of epithelial cells, we would call that an adenoma, right? So the oma part really just talks about the fact that it is some sort of little growth, right? If eventually that adenoma does turn cancerous, that's when you would then call it a carcinoma, right? And really it's still from this adenoma, so it's an adenocarcinoma. So here are some examples of typical adenocarcinomas. Lung cancers, colon cancers, breast, pancreas, stomach, esophagus, prostate, the endometrial lining of the uterus, and ovaries, right? The thing that they have in common here is many of these are tissues that are secretory, meaning they do secrete things, whether it's mucus or hormones or different other types of substances, right? So they have glands involved. So adenomas then are cancers of glandular epithelium. Let's look at an example. So here, let's look at the stomach lining. A typical normal stomach lining, if you were to really zoom in and look at it, you'd see these gastric pits, right? So these little pits are, literally little pits in the lining of your stomach that are surrounded nice and neatly by these different gland cells. So these gland cells secrete things like mucus. They might secrete things like hydrochloric acid that help you digest your food, right? So keep in mind these little pits look like this because we're sort of looking at them top down in cross section, right? So the little space that's been sort of cut, it just looks like it's empty, right? Notice that the glandular epithelium here is nicely organized, right? Around each one of these little gastric pits, right? You have this nice little even one cell thick layer of glands going around them. And this would be all up and down the pit, right? And surrounding it is well-defined connective tissue, and the space in between them is connective tissue. So this would be normal. What does stomach cancer look like? It looks like something like this. So this is a typical gastric adenocarcinoma. I say, what features do you see distinguishing the two here? Right? How does this look different than this? Right? Yeah, why don't you just sort of unmute yourself and just kind of chime in? What are some things that you see are different in the adenocarcinoma versus the normal? Any of you?
Looks like the shapes of the glands have changed. They're not as well defined.
Okay, good. Right, so the morphology of the cells themselves has dramatically changed. They've lost their definition. They've lost their proper differentiated morphology. Right, good. What about the surrounding tissue? Anybody? I don't expect you to have very technical terms, by the way, right? I'm just saying, how would you describe it just by looking at it?
The surrounding tissue looks like more pink.
Yeah, so even the surrounding tissue has changed, right? So what does that tell you? That during the change from normal to disease, it's not just the original cells that are going bad, right? That it has an impact on the surrounding tissue as well, all right? So the other thing you can't necessarily tell very well from this is that the number of these glandular cells that make up each of these pits is now out of control. Rather than having a well-defined number of them neatly lined up, they have just become sort of these crazy kind of piles of cells around them, right? So certainly the loss of control over proliferation, resulting in way more cells than normal. Also the loss of morphology, right? In terms of these cells no longer having their normal differentiated shape and having an impact on the surrounding connective tissue that supports them. All right, good. So those are very important points here. Let's move on. Other examples of epithelial cancers are things like squamous cell carcinomas. So these are tissues, squamous cells are cells that are flat, right? Rather than cuboidal or columnar cells, squamous cells tend to be flat. So these are the cells of your skin or your nasal cavity, the lining of your respiratory tract, for example, or the lining of your esophagus. Well, that's your respiratory tract. And even the lining of different types of organs or surfaces associated with things like reproductive tract, like the cervix. Right? These are squamous cells that form that outer layer. Okay. So here's an example of carcinomas of squamous cells. So this is normal skin on the left. Your typical normal skin, your dermis and epidermis, right, consists of these keratinocytes. So these keratinocytes, the dark purple ones here, are healthy, right? And they form the sort of last outer layer of living cells of your skin, They're called keratinocytes because they make buckets and buckets of keratin, the protein keratin. So once these keratinocytes die, they leave behind this insoluble keratin. And you get a layer of 20, 30 or more layers thick of these keratinized dead cells. And that's your epidermis, right? That's your outer skin that's very tough and impenetrable to things like water, right? So let's look at disease. So this is squamous cell carcinoma. And again, the things that we looked at and recognized on the previous slide with the stomach cancer applies here. Out of control proliferation for sure, right? So here you can see the individual cells on the left, these nice little purple cells, right? You cannot tell anymore where the individual cells are in here, right? There's like a bazillion of them. I use the word bazillion. I get that from my kids, because I don't really know how many cells are in here, right? But there's like way more than you can even see clearly. They've clearly proliferated out of control. There's a change in morphology. And importantly, in this case, it looks like these layers of keratinocytes that used to be very well defined. So this below them, right, these are epithelial cells. So this would have been some sort of a basement membrane that's separating the normal healthy epithelial cells from the connective tissue beneath. Here you see that the epithelial cells are beginning to breach and push their way into the surrounding connective tissue. Okay. So other types of carcinomas include things like small cell, large cell lung carcinoma, hepatocellular carcinomas, so things like liver cancers, renal cell carcinomas of the kidneys, and things that we call transitional cell carcinomas, for example, of the bladder. Transitional cell means different regions of tissue where one type of cell kind of just is not cleanly separated from a different type of cell, but there's sort of this more mixed kind of transitional area, right, of two different types of cells. So that happens in the urinary bladder as you look at the lining, and it goes from places like the ureter into the bladder itself, and then into the urethra, right? All right, let's move to the second type, non-epithelial. Right, so these are cells that are connective tissue, blood, and cells of the nervous system as examples. Right? So about 7% of cancers are hematopoietic in origin, meaning that they have to do with the cells of the blood. Right? Hema means blood, it's Greek. Poetic, you know, I like this, right? Because poetic, just like the word poem, right? It means the creation of something, right? So hematopoietic cells are the cells that give rise to the different components of your blood. So these come from things like erythroblasts. So in other words, the precursors of red blood cells that still have their nucleus, right? Plasma cells, like B cells, leukocytes, like white cells in general. Neuroectoderm cancers, about 1.3%. These are cells that originate in the nerve cells of the central and peripheral nervous system, and other types of connective tissue consist or comprise about 1% of tumors seen in clinics. So these are cells that are part of the surrounding connective tissue, like fibroblasts, adipocytes. So these are your fat cells that store fat, osteoblasts that are involved in the maintenance of your bone, and myocytes that give rise to muscles. Right? So you can see that if 80% of solid tumors come from epithelial cells, that means 20%, right, are not epithelial in origin. And then as we break it down, we see that of those, hematopoietic cells tend to be the greatest portion. Right. So here are an example of some blood-related cancers, including leukemias and lymphomas. So here's a list here. You know, I typically don't require or expect you guys to memorize these lists because I don't like memorizing things. I don't think memorizing is really all that conducive to really learning, but I still present these tables to you because I want you to kind of appreciate and kind of process and think about these things. So these are all examples of blood-related cancers, many of which you've heard of. Right? I mean, certainly you're familiar with the term leukemia and lymphoma, but also myelomas, right, are also here as well. Importantly, these tumors may be liquid, right? So leukemias, because they are out of control proliferation of white blood cells that are circulating through your blood, there is no real solid tumor. And you might think, well, that's kind of weird. You always think of a tumor as being like a solid mass, right? This really disgusting solid mass. But the truth is, right, the term tumor just means some sort of population of cells that's resulted from those cells proliferating out of control. So it might be solid if they're all in the same place, but if you're talking about cells that are already circulating and they're dividing out of control, then it's like a liquid mass, right? So that's what a leukemia is. Or they can be solid, like in the case of lymphomas. So if we're talking about hematopoietic cells that are like in the bone marrow, for example, that are still kind of dividing to give rise to other cells, if you start having tumors form there, right, you might have a solid tumor, lymphoma. Let's look at an example here. This chronic myelogenous leukemia, or CML, right? Because we'll talk about CML a little bit later in the semester. So let's just kind of introduce it here. So here is a typical image of CML. And this is just from the blood. So they took a blood sample from a patient, did a blood smear on a slide, and now they're looking at the blood under the scope. So what you see, these are red blood cells, right? Typical red blood cells. These little things might be, these little specks might be things like platelets, all right? And then over here, you have a typical white blood cell, a normal leukocyte. This might be something like a neutrophil, for example. All right, so here's a normal one, here's a normal one, here's another normal one. Okay, this is what we might think of as a banded neutrophil. But look at these, right? So these are cells, they're white blood cells also, that have lost their normal differentiated state, right? They're really kind of big, they have humongous nuclei, right? So these are undifferentiated blast cells that are now in circulation, right? And they're multiplying out of control. So these are overly abundant and cause disease because of that. Because let's face it, there's only so much space in your circulatory system. And as your blood is flowing through it, your blood, right, should maintain its normal composition of certain numbers of cells of this type, certain numbers of that type. And if all of a sudden everything is just getting filled up with these undifferentiated blast cells, right? Oh, and by the way, in case this term is not familiar to you, typically when you see the word blast in association with discussing cells, it means that it's a cell that's a precursor or that gives rise to something else, right? So blast cells, in this case, are precursors of immune cells that, rather than properly differentiating, are just kind of stuck, dividing out of control, right? As a result, right, your blood is kind of like clogged up with these and interferes with production of normal red blood cells, and the overall functioning of things like your immune system become highly compromised. Right? Not to mention just the normal physiological makeup of your blood and the different functions of your blood, like transporting nutrients and gases and things, can also be impaired. Right? Okay, and then examples of nerve-related cancers. So again, here are some examples. Some of these you may have heard about already. So things like glioblastomas or retinoblastomas, neuroblastomas, right? Again, you recognize certain features of these words. The omas are suggesting that they're tumors, right? If you see things like blastomas, you figure this probably comes from some sort of precursor cell. Right? Let's look at a couple examples of glioblastoma and retinoblastoma. So glial cells are one of the types of nerve cells that you have in your brain. They're there to kind of support the other nerve cells that actually do your mental processing. Okay, so these glial cells are usually kind of star-shaped like this, and they have lots of little dendritic appendages where they can communicate with your other cells, nerve cells in your brain, to help support them. Right? These are cells of your retina. Right? So, you know, in your eye, in the back of your retina, you have these rods and cones, right? So they are arranged in these different layers to do things like capture light and be able to tell colors and things like that. So if this is the normal appearance of glial cells in the brain and the retina, what do cancers look like? All right, so this is glioblastoma. So definitely out of control proliferation, very obvious loss of proper cell morphology. And undoubtedly, just by looking at this, you can imagine that this tissue is not going to function the way it's supposed to function. Look at the retinoblastoma, same thing. These little cells are now dividing out of control. They've not only lost the proper morphology, but that has had a profound impact on proper arrangement and structure of the surrounding tissues. They're no longer in these nice layers. They are just growing out of control all over the place, right? And I don't think it takes a lot of imagination to realize that these cells no longer can function properly. And things like blindness is almost inevitable. Okay, so this slide just says that connective tissues that arise from things like connective tissues associated with fibrous cells of your connective tissue in your body underneath your epithelial layers, or things like muscles, right? We tend to call these sarcomas. So sarco, you probably remember, because when you're talking about muscle cells, they have sarcomeres, right? That's how the muscle cell functions properly, part of its normal shape. So whenever you see sarco as part of a identifying tag for a tumor, you know, oh, it's connective tissue, and it may actually be some sort of muscle related. Right? So here you have things like fibrosarcomas, chondrosarcomas. So in this case, this comes from chondrocytes, which are the cells that make your cartilage. Right? Adipocytes give rise to liposarcomas. Right? So these are fat cells that have gone bad and have become cancers. All right, so let's look at this leiomyosarcoma here, which is a smooth muscle cell sarcoma. Remember, you have two types of muscle in your body: the skeletal muscles, that are the ones we typically think of when you think of muscles, right? Your biceps, triceps, whatever, that help you move around because they're attached to your skeleton and help you move your limbs around. But you also have smooth muscle, the muscle that lines things like your GI tract or even all of your blood vessels, and is in charge of doing things like constricting or relaxing different blood vessels or your intestines, right? So that you can do constrictions that move the food along. So let's look at leiomyosarcoma. So this is a smooth muscle sarcoma. In cross-section, we see on the top and bottom of the slide, very typical healthy smooth muscle. So healthy smooth muscle is not striated or striped like your skeletal muscle is, right? Smooth muscle has this typical appearance and you have the little nuclei of each of the smooth muscle cells kind of shoved over to the side here. Right? And then you have these big bundles and groups of smooth muscles working together. This circular thing is actually the cross-section of a blood vessel that's running through the smooth muscle. Right? An important thing to keep in mind is that all of the tissues in our body have to have a network of blood vessels going through them to supply the living cells of the tissue with nutrients, get rid of wastes, and do things like gas exchange, oxygen, CO2. So this is a cross-section of one of these blood vessels flowing through here. This is the tumor. And you can see this is a band of proliferating cells that's kind of running through here. They've lost their normal morphology. You can imagine this thing's no longer going to function properly as muscle tissue. And what is particularly scary is that you can see these cells are also spreading, right? You have this impression, like especially here, right? That it's not really just confined to this one area, that these diseased cells are beginning to kind of infiltrate the surrounding thing. And what's particularly scary is some of them are making their way up against this blood vessel. And if they breach the blood vessel and enter the blood, then all of a sudden they're whooshed away and carried to other parts of the body, which we can call metastases. Right? So the last type of three main types are mixed lineage cells. And this is when some tumors don't fit very neatly into epithelial or non-epithelial because they have ambiguous developmental origin. So in other words, some things like melanomas You might think, well, what are you talking about? Melanoma is like skin cancer, right? So doesn't that come from epithelium? No, because melanomas don't come from the keratinocytes that make up your outer skin. They come from the melanocytes, right? These are special cells that kind of hang out, it's sort of just under the epithelial cells and do things like make melanin, right? The brown protein that gives you a tan. Right? We actually aren't entirely clear where these melanocytes originally come from as the body is developing and these different tissue layers are forming, right? So that's why we call it a kind of ambiguous developmental origin. And certain types of small cell lung cancer fall in this category as well. Right. Okay, next, let's talk about the embryonic origin. This idea of being able to trace back the original primordial or early germ layer that these different types of cells come from. I don't have a ton to say about this. So even though we're getting on in the lecture here, it's not like you're like, oh boy, we have to like listen to a whole bunch of other stuff here. All I wanted to say is that as the fertilized egg is developing and dividing and forming a blastocyst, right, from 1 to 2 cells, then 4, then 8, then 16, then 32, et cetera, once it gets to the point where it is several hundreds, if not thousands of cells, then the development begins to change. And it's not just about proliferating to make more cells. It begins to be certain groups of these cells start changing and going down different differentiation pathways to become the different types of tissues in our body. So you can see here in this stage of embryonic development, in cross-section, they've used different colors to kind of suggest that these cells that are parts of these different regions are now undergoing different programming, right? They're making different decisions about differentiation pathways that will lead to things like the mesoderm that's going to form things like the epithelial linings of things like muscle. Then you have the neural ectoderm that's going to form the nervous system. You're going to have the endoderm that's going to start forming the inner linings of things like your organs. Right? So as the embryo develops further, you can see here they've taken a cross section, right? So these early decisions on the left, ultimately, which are driven by genetic programming and different genes turning on at the right time, will give rise to these different layers: skin, muscle, nervous system, and the gut. So why is this important? Because once you have an idea of what the original normal cell type was that gave rise to a cancer, right? Remember, the whole point of doing that is you want to have some insights into what cell signaling pathways are the most relevant in that normal cell type that might have gone wrong to form cancer. What this is talking about is taking it a step back even further. As we will learn, during the development of cancer, many genes that should be turned off in fully differentiated cells in our body, right, and that have been turned off for many, many years, right? So for your early few weeks of development as an embryo, these genes are things like early developmental genes that help define the different primordial germ layers that are forming. At that point in your early development, these genes were active to help define things like body plan, right? Symmetry of the body that's forming, where things like limbs are going to be, where the nervous system is going to form, or the organs of the gut. By the time you're, 20 plus years old, like all of you are, right? Those genes have long switched off in the fully differentiated cells in your body. But remember, they're still there, right? One of the most important concepts here is that every cell in your body, because of this genomic equivalence concept, has the same genome in it. So even though your skin cell that you see on the outside of your body no longer uses some of these early developmental genes, the genes themselves are still part of the genome and may be improperly switched on and could wreak havoc if they're switched on, right? So that's why understanding the original primordial germ layer that different tissues came from, again, gives you another level of insight into understanding, what are some of these critical genes that may have accidentally got switched on again as part of the development of cancer, right? And do we have different types of drugs or other therapeutics that can target those specific gene pathways? So consider that the cell type fate, this whole process of differentiation, depends on emphasizing certain gene pathways over others, right? And again, this is just all related to understanding the specific origins of a particular tumor. Okay. And then the third category is biological behavior, whether the tumor that you're studying from the biopsy, is benign or if it's malignant. So what do we mean by a benign tumor versus malignant tumor? Right. I mean, it's a pretty simple functional definition. And I think you already, even though this is the only second day of class, if I asked you what's benign versus malignant, you'd probably have a good sense of it, right? Benign is when tumor cells stay local. They don't surround invading tissues, right? It's just like these guys, right, hanging out in their little man cave watching the game, right? They can get as crazy as they want in their sort of like latent frat brother kind of mentality here, right? But it's okay because they're not causing anybody any harm, right? They drink their beer, they eat their chips, and they high five each other and do awkward, chest bumps or something. But if they get out, right, and get into the public, so like at a rowdy sporting event, right, that's when they can cause trouble to surrounding cells, right? So that's malignant. So when we're talking about a tumor that stays well-defined in its well-defined space, right? For example, at the spot that it originally formed and it just kind of stays there, then we say it can stay there potentially indefinitely and it's benign. It really doesn't cause any harm. But if those cells that make up the tumor continue to change, right, and that process of continuing to change, we will be studying a lot this semester. What happens is eventually those cells get rowdy, right, and they break out. And if they spread into surrounding tissues, or like I showed you before on the slide, oh boy, if they make it into the blood system and have the freeway to go through the body, then they can become malignant and start spreading the cancer or the tumors to other parts of the body. So a benign tumor is not cancer. Cancer is when the benign tumor has changed and is spreading and become malignant. And therefore, that is now cancer. Okay. So here is quickly some definitions of how we describe changes in morphology of the tissues. Normal tissues, for example, this is a cross-section of, I think, the small intestine. All right, so this is the lining of the small intestine. These very columnar kind of shaped cells here are the epithelial layer. This would be the lumen of the intestine where food would pass, right? And then the other side here, on the other side of the basement membrane, would be connective tissue where the nutrients end up being absorbed by these normal mucosa. This is an interesting slide because what they've done is they've taken the sample of an adenocarcinoma here, right, and made sure to still capture some of the surrounding epithelium. So you can actually see a transition happening, right, from the normal mucosa. So on the very upper left here, you can see the well-behaved, well-defined glandular epithelial cells. And as you move down past this dotted line here, you see that things are beginning to change. They're beginning to lose their normal shape, right? So we call that plasticity, right? Plasticity, plastic. Plastic means something that's very malleable and variable in shape, right? So something that is dysplastic means the shape is beginning to change from normal. And you can see that here, right? And then eventually, as you follow this around, you can see that you really have the telltale signs of out of control proliferation and growth and alteration to the surrounding tissues, right, where you get into the full-blown adenocarcinoma. And then metastases, right? So we just said that metastases is when the cells from a primary tumor site that might have been a benign tumor have changed to the point where they then learn how to escape the confines of that original tumor site and are now spreading. Right? So I know that this is a really gross looking slide, but it's hard to avoid looking at gross slides when we're studying cancer. Right? Here we have a healthy lung from a mouse versus a lung from a diseased mouse where melanoma has spread to the lung. So again, melanoma that started in the skin from the melanocytes have now spread and caused the disease that you see here. Here is an example of a diseased liver. The white spots that you see are colon cancer cells that have gone from the colon, so the lining of the large intestine, and have found their way to the liver and are now forming these new tumors. Right? And then this one on the right is an example of brain cancer, right? But importantly, right, it's not brain cancer that started off as brain cancer. It did not start off as glial cells of the brain turning into glioblastoma. Likewise, the liver cancer that was diagnosed here actually turns out not to be true hepatocarcinoma. It's not really liver cells, right? These came from other types of cancers. In this case, breast cancer that spread to the brain, right? So you can already appreciate this whole idea of how complicated it is to be able to tell where a particular cancer cell came from, and yet at the same time, why it's important, right? Because if on the right here, this were a true glioblastoma that started off with nerve cells of the brain going bad, right, then you might have different chemotherapy or therapeutic drugs to target those types of tumors compared to if the tumors actually started off as breast tissues. Okay, so this just kind of wraps up and brings us back to this again. So what I encourage you to do is take a look at this, right? Don't worry about memorizing it. Look through it, talk through it, and see if the way this is organized is consistent with what we discussed today and how these different categories are divided up. Once you kind of are comfortable with that, then you begin to understand, right, that this is the type of challenge that a clinical oncologist faces. When you have a patient who presents with disease, they send like a little biopsy of the tumor to the pathology lab. The pathologist takes a look, right? And what they're trying to do is they're trying to fit it in somewhere on this chart, right? To get that valuable insight as to what types of drugs are probably the most directly effective in treating this person's cancer. Right? Okay, let me go ahead and stop the sharing. And I'll stop the recording.